So I'll start with some principles of modeling of cancer initiation and describe two types of mutations: loss of function and gain of function mutations that behave very differently when you try to model them mathematically. We'll talk about the rate of evolution. So my main kind of application is the role of aspirin in colorectal cancer. So that will be my biggest application. And if we have time, the last topic is kind of the hottest one for me right now, what I'm most excited about. It's unpublished. It's about some scaling laws in spatial evolution. Let's see if we get there. If not, tomorrow. So this diagram shows the steps to colon cancer, starting from normal epithelium. The first event is thought to be the inactivation of the so-called APC gene, followed by an oncogene and some other modifications leading to late adenoma and then carcinoma, a stepwise process that has been studied a lot, characterized now for a long time. But I want to attract your attention to two events that happen here, which are very typical patterns for many cancers. The first one is an inactivation of a tumor suppressor gene, and the second one is an activation of an oncogene. So these two types of genes, there are many, many members of those families. So in colon cancer, it's APC and KRAS genes and others too. Let's create models of how evolution progresses if it involves one of these. So start with an activation of an oncogene. So this is what they call a gain of function. A mutation occurs which creates some new property in the gene, which presumably increases the fitness of the cell, and that cell has a probability to invade the population. So one event happens, and so the properties of the cell change and it becomes advantageous. Very different picture arises when we're dealing with a loss of function mutation, which is a tumor suppressor gene. So here, say the APC gene is there in place to prevent bad things from happening, right? So for example, it prevents unwanted reproduction of cells. And this gene is presented in two copies, like most of the rest of the genes. You inactivate one of them and nothing much happens because the other one is still there to prevent bad things from happening. So you have to really inactivate both of them. So it's a two-step process. And what happens to this intermediate cell, which with only one cell, copy of the gene inactivated, it's not the same cell, right? It doesn't have the advantage like this one. This one has a larger fitness. This one may have a disadvantage because the gene does other things. And if you mess it up, so maybe other functions are not working properly. So the fitness of this intermediate cell can be the same as the wild type or smaller, typically. So I'll present kind of older work where we characterize these, and then I'll transition to applications. So how do we model an evolutionary process? The simplest process is what they call Moran process. It's the least realistic of all of them. And basically you have cells in a box. You don't keep track of where they're located. And at each moment of time, you kill a cell and you reproduce another cell such that the population stays constant at all times. So what are the possible outcomes of this process if I have two types of cells? So either the green mutants die out or they take over. So in this process, we can calculate the probability of both of these outcomes. It's a very standard problem in probability. Let me mention that in this picture, there are two types of cells, which are denoted by different colors. We can assign fitness to them. And when we choose a cell for reproduction, we do it based on the cell's fitness. So if a cell has if mutants have a smaller fitness, a parameter than the wild type cells, they're slightly less likely to be chosen for reproduction. So we can derive the probability of increase or decrease in the number of mutants with each step, and then calculate the probability of mutant fixation. A less standard problem that we tackled a long time ago is what is the probability that by time t, a 2-hit mutant has been created. So now it's a 2-step process, and you kind of go from cells that are normal. Maybe some of these are created. The fate of these cells is uncertain, because if they're disadvantageous, they're more likely to die out than not. But perhaps before they die out, the second copy of the gene is inactivated, and then you have a winner, right? You have a supercell that expands. So this is this type of process. So let's look at the Moran process associated with this. So we go until we create one of the supercells, and we stop then. So how long does that take? The answer to this question depends on parameters, but this is such a simple model, not so many parameters are there. So what are the parameters? There's a first mutation rate, the second mutation rate. These are typically known for inactivation. It's usually, I assume it's 10 to the minus 7 per cell division per gene. Another parameter is this, and this is important. So this is the fitness parameter that characterizes the intermediate cells. So if we plot fitness as a function of the number of mutations, we start from the fitness of the wild type here, and there could be a slight decrease in fitness, and then the fitness of the super mutant is much larger. So this is kind of when bad things start happening. So this is a fitness landscape from wild type to advantageous super mutants. If you flip that, this is an energy barrier, and if you're a physicist, you recognize that. So to go from here to here, you kind of have to overcome an energy barrier. So this is a very nice transition between biology and physics, which is accomplished by flipping this fitness landscape. To make the long story short, you can calculate, and going back to physics, right? To go from here to here, you have to overcome this barrier. And the term is that is often used as tunneling. You have to tunnel through, right? So we call this stochastic tunneling in biology, where you go from here to here. And the fact that these mutants are either neutral or disadvantageous makes this process very long, right? Each mutation rate is very small. The population of these cells is very small typically, and they keep dying out, right? You have to kind of accumulate enough of them to create the second mutation before it can expand. So that's what makes it long. So this is kind of like a rate limiting step. So to go from here to here can serve as a proxy of the evolutionary rate. How quickly does evolution progress? So we can calculate this rate in terms of the parameters of the system. I skipped this, I think. This is the methodology that can be used. And the point is that, for example, for neutral intermediate mutants, where they have the same fitness as these, we have this formula that involves the total population and the 2 mutation rates. And if this is disadvantageous, the disadvantage comes in the denominator here. So we have a formula. Good. So that was a non-spatial model of cancer. So can we make it a little bit more realistic? So the next step is to add space. And this is the simplest way to add space, is to look at 1D. This process now is like this. When you kill a cell, only one of its neighbors can reproduce. Again, it's a constant population process, but now competition happens locally, which changes completely the evolutionary processes here. So I think this work is like 2006, 2007. I'm still extremely interested in evolution in space. A lot of very counterintuitive things happen. So this is kind of the first attempt to tap into that, right? So let me tell you a bit about this. So, okay, again, we stop when a super mutant arises, and we can calculate the rate of evolution in this case. This is the formula, for example, for the neutral intermediate mutants. And the point is that it's faster. So adding spatial interactions, making competition local, speeds up evolution in this case. And you can see why it's, so you have to compare this formula with that was derived for the non-spatial process. You can see that this small number, we take a square root, and here we take a cube root. So when U2 is very small, this is a lot larger. Okay, so you can prove that space speeds up evolution tremendously. How about 2D? Right, so 1D cancer has limited applicability. 2D, 3D is more realistic, but basically the same thing is observed in spatial 2D and 3D models. And in fact, so what you could do, you can look at various sizes of the neighborhood. So as I said, in space, evolution happens locally. You only compete with your neighbors. So you can compete with your closest neighbors, or maybe the second closest. You can expand the range at which you kind of sense other cells and you compete with them. The larger the range, the closer you are to the non-spatial simple Moran process. So the effect weakens. But if you have some neighborhood, again, the evolution is sped up significantly. There is a simple reasoning behind this, which I really love how intuitive this result is. So here I want to demonstrate two numerical simulations that kind of explain all of it. So on the left, we have a simulation where red is normal cells and light blue are single hit mutants. So these are those weak cells that pop up, they come and go, they are usually outcompeted by the wild type background. And this is a non-spatial simulation. So even though it's presented on a square, we don't care where the cells are. Each cell is competing with everybody else. And this is a spatial simulation where when you reproduce, you can only reproduce next to you, the 8 cells in the neighborhood. And you can see, of course, now the mutants, which are quite rare, tend to occur in islands because they reproduce next to each other. And now here's an explanation of why evolution happens faster in space. Whoop, what's going on? Sorry. So here, each of those mutants is competing with everybody else. And the mutants' prospects are grim because the fitness is lower. Here, the mutants are competing with their neighbors. So if I'm a mutant and I'm surrounded by other mutants, I feel pretty good because I'm not weaker than my neighbors. So I hang around longer because I'm not being outcompeted. So mutants that cluster in these islands survive for longer. So they have a better chance to create a super mutant, which requires one more mutation. So this clustering in space serves as a mechanism by which evolution progresses faster in space. Okay, and the last structure that I want to explore here theoretically is adding another bit of realism into the model. So before we looked at all the cells as kind of particles almost, right? But in biology, we have to remember that often cells are arranged in a hierarchical structure. My yesterday's lecture was all about hierarchical structure. So here, let's see what that arrangement does to evolution. So this is the simplest model of a hierarchical structure. You have a stem cell at the bottom, and it, say, divides asymmetrically, creating a more differentiated cell. This one doubles, these double, and so on. The last row of cells are the most differentiated cells that perform their function and die through programmed cell death by apoptosis. So when they die, these guys divide, these divide, this divides, and this divides, right? And the mutant can spread. You can see the blue mutant, and so on. So we can repeat the calculation of the rate of evolution in this case, and it's slower. It's slower than the mass than the spatial system, but it's also slower than the mass action, than the most primitive one. This is the formula that has to be compared with that, and u is small. Take the limit to infinity, and so here's the picture that transpires here, we can plot the rate of evolution as a function of the mutation rate. And here in the middle is the first most primitive Moran model, non-spatial. This one is a spatial model which accelerates evolution. And here is a hierarchical model that decelerates evolution. So the thinking is that perhaps our organs often are built in a hierarchical manner. It makes sense from the point of view of evolution. We evolved this way because the hierarchical structure slows down the advancement of the generation of cancer. So it's advantageous for multicellular organisms to be characterized by this hierarchical structure. And even though here I only showed you the most primitive kind of binary division model, we also have explored more realistic models of hierarchical structure with the same result. Evolution is slowed down very significantly in those models. So, this slide demonstrates a variety of hierarchical organizations of tissue. So yesterday I talked a lot about this one. This is a hematopoietic system. Today I will mostly focus on colon cancer, which is also organized hierarchically. And now I want to apply some of this to a very kind of applied problem in collaboration with experimental colleagues. And in this problem, we looked at the role of aspirin in the delaying of the generation of colon cancer. And to me, the kind of interesting part about this project is the connection between the microscopic evolution that happens as a stochastic process in small compartments of the body and the epidemiology of cancer. So aspirin kind of affects how quickly cancer develops in the population, right? So here we provide a link between the two very different scales. So colorectal cancer is one of the most common cancers in the United States and in the world. It's the leading cause of cancer-related deaths. Many new cases have been diagnosed, and many people die every year. On the positive side, the colorectal cancer rates have been steadily decreasing in the adult populations because of colonoscopy and other prognostic tools. Kind of on the negative news, higher rates of colorectal cancer have been observed very recently among young people, those who are younger than 50. And actually, I think nobody knows why. So that's a development that was quite recent and quite alarming. So how do we model it? So we are going to focus on the first two steps in this progression. So it will involve the inactivation of a tumor suppressor gene and an activation of an oncogene. Let me first describe the very first step, the APC gene. So in a sporadic colorectal cancer, this really is two steps, as I already explained. You go from a functional APC gene to one copy inactivated and then both copies inactivated, a two-step process. And this is the step that takes the longest in the progression, decades. It could take 20 years, right? There are familial conditions that make this shorter. So some people, unfortunately, are born with this first mutation that they inherit in the germline. So they start here. And this is called FAP, familial adenomatous polyposis. And this population develops colorectal cancer in their teens, and nobody survives much longer. So that's one of the familiar conditions. And I want to mention another familiar condition. So, and for that, I have to talk about microsatellite instability, which I will come back to later in the talk. So this is the straight pathway to cancer from here to here through these two mutations. Another pathway that happens in 13% of cancers is first to inactivate one of the MSI genes. So these are the genes that control the amount of errors that you receive, that cells receive. So if a mistake is made, it has to be checked. If you notice, correct it and go on. So this is something that helps reduce the mutation rate. And there are genes that are responsible for that. If one of those genes is inactivated, your error rate is going to be a lot faster. So, sorry. And this is again a two-step process to go from here to a state where the mutation rate is a lot faster. It takes 2 mutations, but once you're here, it goes really fast, like 100-fold faster. The mutation rate is increased significantly. So you can go from here like this, or through inactivation like that. 13% of the patients have this type of instability. A familial condition, another familial condition is what they call the Lynch syndrome. People are born with one of those copies sorry, I think this is a mistake. They're born with one of these copies inactivated. So they only have one step until this fast pathway. So this is not as deadly as the previous one that I mentioned. But the population of these people usually develop cancer in their 50s and 60s at a much higher rate, and mostly colon cancer, but other conditions as well. So this will be important for us. So when we talk about aspirin, I want to first describe experimental observations. There was a huge clinical trial performed by John Byrne in Great Britain, where they administered aspirin to a large cohort of patients and compared the rate of Lynch syndrome cancer in a huge cohort of patients. So these were not regular people. These were people born with this macrocyclic instability gene inactivated. So they are predisposed to develop colon cancer. And you can see here, this is placebo, and this is the red is aspirin. The incidence of developing colorectal cancer was significantly reduced in those who took aspirin. And this effect was observed for some years, right? And there were several trials like this. Here is a meta-study that went through many papers that studied the role of aspirin in colorectal cancer. And so these are the different studies. And this is what they call the risk ratio. Risk ratio is the risk to develop a condition with treatment divided by the risk without treatment. So if this is less than 1, it means that treatment or prophylactic is effective, right? So, and you can see that in all of these 8 studies, the risk ratio is less than 1. So a lot of evidence that aspirin helps. But the study that's most important for us, and I want to concentrate on this one, is a study by Andrew Chan and group. This is not Lynch syndrome. These are regular people. This was a huge study of nurses. This is how many participants they had, 10,000 participants. And it's not a controlled study. It's a survey study. They asked people, how many aspirin pills do you take per week? So there are several groups of people, 0 pills per week, or 14 pills per week is the largest dose. So 14 pills is two pills a day. So take it for headache, for other reasons, right? So there's a spread of people who do that. And then they looked at the incidence of, that was not colorectal, fully developed colorectal cancer. It was late adenoma. And I'll tell you exactly what it is. It's a pre-cancerous state. So you can see that for the largest dose, the risk ratio is 1/2. So very similar. So the probability to develop this condition was decreased by a factor of 2 in these people. So nobody really understands why that is. So we tried to kind of figure out what could be some of the reasons for this. And this is a study that has been going on for several years, for many years, with 2 experimental collaborators, Ajay Goel from City of Hope and Rick Bolland he is now retired from UCSD. And this is work with Dominic Wodards and a former postdoc, Yifan Wang. So first, I'll review the experimental results that were obtained by our colleagues. So they injected cell lines into mice. So these are xenografts, the xenograft study. And they collected the tumors and they exposed it to different various doses of aspirin. So these are some of the photos of the tumors. This is the amount of aspirin that was used. And we fitted a mathematical model to describe the growth of those tumors. So these are 3D tumors. So the model is like this. The volume grows proportional to the surface. So we often see here the non-spatial models of cancer. If you want to do it in space, it's a little bit different. It's not an exponential growth. When you have a three-dimensional sphere and it expands, when cells divide and die inside, that's not going to increase the volume because it's just inside, right? So the only way you increase the volume is when the cells divide on the surface of the sphere. So the growth rate will be proportional not to the volume, but to the surface, which is volume to the power of 2/3. So you can solve this very simple ODE, get your growth law and fit that. So we were able to actually estimate both division and death rate of cells in this model. And the second study, actually the first study, was done in vitro. So I'm doing it in the wrong order. So they first did it in vitro with cell lines, and then they did xenografts. We did both. In vitro, it's exponential growth. In xenografts, it's a 3D growth. We did both. And so this is the result of the measurement of the death rate of cells and division rate of cells as a function of the aspirin dose. You can see here, as the aspirin dose increases, the death rate also increases. The 4 different lines correspond to 4 cell lines that they used. And the result is the same in all 4. Now let's look at the division rate. As the aspirin dose increases, the division rate decreases. So these are kind of mechanistic effects of aspirin that it has directly on the cells. It makes them die more and it makes them divide less. Okay. And we were able to characterize by how much, right? You can see what the effect is as a function of aspirin dose. In vitro, same thing. Okay. So now let's look at the incidence of advanced adenoma in the general population. So these are data from some epidemiological study. This is not the study of nurses, it's a general population, no aspirin. This is the incidence as a function of age, a standard rate age incidence curve. You can see 3 curves here. Red is women, blue is men. We don't know how to model the difference, so we take the average. So black is the average. And we know that as a result of aspirin, this goes down by about 50% in the case of the strongest dose. Can we explain that? So this is the question we asked. With the measurements that we did that characterize the effect of aspirin, can we explain this reduction in the rate of incidence of advanced adenoma? Okay, so we created a model of cancer generation, parameterized it by using the measured values, and try to see what happens. So let me explain the steps. So colon, we have to consider colonic crypts, which are small folds of tissue. They're microscopic. Each of these folds is about 10 to the 4 cells altogether. There are 10 to the 7 crypts per colon. Stem cells are located here at the bottom, and we're going to focus on stem cells only here. So we're not going to worry about mutations that happen later on because presumably they get washed out and they are not consequential. So we are looking at the population of stem cells. So the differentiation takes about a week. So just to give you an idea of this thing. So we look at the transformation of cells from wild type to mutant, and we take account all these steps, right? The tumor suppressant gene, oncogene, these things, and then yeah, there's a lot of theory that goes on here, and this is the main point. So what is advanced adenoma? So I talked a lot about the inactivation of the APC gene. So you go from APC++, APC minus mouse. So that's inactivated. That's not late adenoma yet. One more step is an activation on an oncogene like this. So first APC and then oncogene KRAS, the combination of both lead to the state of advanced adenoma. This is what was studied in the nurse studies. Right? So this is our data. So advanced adenoma is here, but of course there are different pathways to go from here to here. The one that I described is first an activation of the APC gene and an activation of a tumor oncogene, or you can go like this: first oncogene, then tumor suppressor gene, or like that, right? So there are 3 different pathways. We include them all. It's a stochastic model. Another thing that was important to include, and it's something that only became apparent relatively recently, once a crypt is transformed, for example, it has both copies of the APC gene inactivated, it undergoes this weird process of crypt fission. So the fold, the fold of cells, like the crypt, multiplies, duplicates itself, and creates like a whole population of clones of that crypt. And it happens with all of these, not the wild type, not this one, not the intermediate one, but everybody else undergoes this process of crypt fission. Okay, so this is something that is known. So the model, this model first appeared in this paper in 2020, which made us extremely happy, because we tried to do it for years, and there were simply too many unknown parameters. It was not possible to parameterize a model in any reasonable fashion. I felt like we could predict anything. So after this paper, we were able to learn a lot of the parameters. They have been measured, and now we could parameterize the model. So we modified this model. So this is a model that operates on the level of crypts. N1, for example, is the number of wild type crypts. N2 is the number of crypts of this type. And this is the average, the expectation. So we go from the stochastic model to the expectation of the number of these crypts. Okay, we modified the model. Oh, and so I have to mention that. So a lot of parameters here are now known. So this is nice. And we added some bits to the model in order to be able to fit it to the data for the epidemiological data. So this is a logistic growth term. So when crypts undergo crypt fission, they shouldn't multiply exponentially forever. We felt like this process has to be capped. So this is a reasonable assumption. In fact, without this assumption, we showed that you could not really fit the data. And there is no aspirin yet in this model. This is just generation of late adenoma. So we fitted the model for some of the parameters that are still unknown. So some of the unknown parameters are the rates of crypt fission. So these are 2 parameters, and the carrying capacity in these terms. So we were not able to get like unique parameter values for these 4, but we got a parameter region where the error is small. So whatever we do, we always do in the region of small error. And where exactly you are in that region doesn't matter. Okay, so now what do we do about aspirin? We take this model and we assume that the dynamics proceed from birth to a certain time, at which stage we apply aspirin, and then we measure the probability that late adenoma has been generated. And what does it mean we apply aspirin? Applying aspirin means changing the parameters of the model. And the way we change them is we translate our mouse studies into human parameters. So it was news to me, but there are tables out there that kind of translate all the parameters from mice to humans. So the aspirin dose that was administered to mice by our colleagues, we were able to translate it into the number of pills per day for humans. Okay, and so there are two types of changes that we implemented in the model that characterize the action of aspirin. First of all is the transition rate. This is the rate that I told you we calculated a long time ago. It's based on parameters that changes as a function of aspirin dose. And the second thing that changes is the rate at which these crypts undergo crypt fission, because this ultimately, this is also a process of cell division. So this changes as a function of aspirin. And so these are some of the results. So we apply aspirin for a certain amount of time, and we measure the effect at a later stage. So for each of these graphs, the fat black dots are the data, epidemiological data for a general population, the rate incidence for late adenoma. The thin black line is our model fitted to that data. Okay, it fits. That's how we parameterize it. So we can reproduce the general population. And the yellow line is the population that took aspirin in our, this is simulated aspirin. And so the blue rectangle is the time span during which our artificial patients took aspirin. So for example, here, it was administered between years 20 and 30. Here, these patients took it between years of 30 and 40, and so on. And the yellow line tells you the risk of developing cancer for those patients that took aspirin. And you can see that there is a significant difference in rate incidence from the epidemiological data for regular people, general population, and those who took aspirin. This difference is significant, but it decreases with the age when the aspirin is taken. So if you take it late, it still makes a difference, but a much smaller difference. The biggest difference is observed when you take it earlier. So we were able to classify this completely. So dose dependence. You can administer a strong dose, 14 pills a day, 14 pills per week, medium and light dose. So this is the relative adenoma risk for these patients, depending on when aspirin was taken. So it is, of course, the largest reduction for the biggest dose, but it still is less than 1, even for light dose. So these data were not possible to obtain from the study of nurses, because even though it was a large study, when you split it all these different ways, you completely lose statistical significance, right? If you want to split the population into people who took it for between years of 20 and 30 from those who took it, so you lose it, right? So this model that was calibrated kind of gives you an ability to play with these parameters and to see the effect depending on these various modifications. So length of treatment, right? How long do you take it for? 10 years? Do you take it for 5 years? So here it shows that, of course, the effect is decreased if the length of treatment decreases, but you still have a significant effect even if, after 5 years of treatment. So I think, so this is it. So for this project, I just want to emphasize that this, when you talk about aspirin, right, it's a complex process, and the effects of aspirin are multifold. This is the most primitive way to look at aspirin. We don't look at inflammation, which is like the most obvious thing, the aspirin reduces inflammation. We don't look at its mutagenic process, the influence, right? Maybe it increases or decreases the mutation rate, who knows? All we do is we look at cells and what they do under the influence of aspirin, division rate, death rate. So these straightforward, measurable quantities translated into humans lead to the effect that is compatible with what is observed experimentally. So this is a kind of first start. And this can be improved by looking at these other processes. So this is kind of like the first step. So now I think I have time to talk about this, laws in biology. Right, I have 20, yeah. So this is less practical, but kind of fun. So laws in biology is ambitious. So I'm going to talk about scaling laws. And I want to ask your opinion about what you think later on. Maybe you can have some comments. I'll be happy to hear that. So I already showed you some laws that we derived. This has to do with the rate of evolution as measured here. So remember, we did it in the basic Moran process. And for neutral intermediate mutants, we get the rate that is square root. We add space, that's cube root. Rick Durrett did it in 2D and 3D. We added hierarchical structure. So you get a different formula depending on the geometry, dimensionality of this process, and other structural changes. Those of us who study PDEs, they know that dimensionality often defines many things, very fundamental. So here in evolutionary biology, the role of dimensionality of space also seems to play a significant role. So these are scaling laws that are dependent on exactly the spatial organization and other things in the model. What other scaling laws can we think of? So I'll go over this one very briefly. This is a simple system of expanding cells or organisms that are increasing in two-dimensional space, which mimics the process of invasion. Of course, tumor is invading. Other things invade too. This is bacterial growth. This is some invasive plant that grows out. So what are some of the laws of invasion? So in this most primitive thing, we design an ABM, or agent-based model. Cells divide and die. We start from the middle of the grid. They grow out. They have division rate and death rate. How do they grow? They grow, and then they saturate if it's a finite grid. So you can get this. So I wanted to calculate the density of cells in the middle of this. So this is kind of, in cancer terms, this is your necrotic core, or like the middle of the population. What's the density, right? So we derived the laws of this density depending on the type of grid that you have. So that's a scaling law, but I didn't want to focus too much on that. I want to add mutants now. So we're here to study cancer. We're here to study the evolutionary biology of cancer. So mutants are very important. Imagine the same expanding population, but now we add mutations. So in this simulation, in yellow, you can see a mutant. Many colonies of mutants. As the colony increases, the mutants kind of form these interesting looking slices, pizza slices. They kind of grow together with the population. The question is, can we predict the amount of mutants that are contained in a growing population, depending on the geometry? These are experiments. They look better than the simulation, right? Which is kind of amazing. Look cleaner almost. And this is done in yeast, a couple of them. These are very important works by Halachek and group that kind of started this whole business. These are experiments in bacteria, but you can see that these pizza slices is not a figment of my imagination. This is what happens, right? So these are simulations that can be performed for neutral mutants. They grow like this. Advantageous mutants make thicker slices. They kind of expand like this. Disadvantageous mutants don't form slices. They form bubbles. They are disadvantageous, so they kind of get closed up. So can we predict how many mutants are going to be there in the population? So we did that, and these are scaling laws for these things. but this was before we understood it, right? So we kind of reported the results. Now we understand it completely. And I want to show you some very beautiful laws that we were able to derive. I think they're beautiful. So first look at a population. So I was always showing you circles expanding outward, right? So let's do 1D because it's easier. Let's suppose that we have like a slice of cells and they grow in one direction like this. And let's suppose we have neutral mutants, and the mutant kind of occurred here, and it keeps growing with the population. Because it's a neutral mutant, you don't expect this thing to expand or on average or contract. It's just kind of going to be the same. So you can calculate how many of them you can expect depending on the mutation rate. For advantageous mutants, they each expand. So you can derive a law for that. Now we go to 2D circular geometry. Similar way, we derive the laws. And look at this beautiful result. So we have 1D, 2D, and 3D. Wild type cells, so without any mutants. What is the growth law? In 1D, a stripe of cells will expand proportional to time, right? It just goes this way. It's not an exponential growth. It's a growth that is linear in time. A circle grows proportional to t squared. That's this. In 3D to t cubed. I already told you that when I explained about our fitting xenografts, that tumors growing through their surface. Okay, so this is well known, surface growth. Now let's look at the law of the growth of neutral mutants. For 2D, they grow as t cubed. And for 3D, they go as t4. You see, the power of time is added by 1. They grow faster, even though they're neutral. And advantageous mutants grow even faster. One more. Okay, so it's a very kind of very logical thing. So I don't want to dwell on details. We derive these laws and we check them with computer simulations. Okay, it all works. We want to look at double hit mutants. So these are simulations that show double hit mutants. So you can see yellow inside yellow. These are the secondary mutants that appear there. Can we predict how many of these are going to be in a growing colony of cells? So to make the long story short, this is the law. It may look a little bit ugly, but it's an analytical expression that, well, this is the most general thing, right? So basically, this is the result. So we're looking, let's see. We're looking at a number of mutations. It could be one mutation, double mutant, triple mutant, and so on. We can arrange how fitness properties change. So for example, during the first mutation, the fitness goes up. Maybe it doesn't go up here, but at this stage, it goes up again and goes up again, right? So we have a chain of mutations with given fitness properties, and we have given mutation rates at each stage. I claim I can tell you how many of these mutants are expected to be observed in a colony of a given size. What is the scaling law? These are the scaling laws. So for example, if I want to know, sorry, this is covered, single mutant, so that's n-1, that is mutant, that is neutral in 2D. So I look here, and the growth law is, you have to go here, 3. It grows as the radius of the colony to the power of 3. Radius of the colony and time are the same, so it goes as t cubed. If I want to go and look at a single mutant that is advantageous in 2D, so single mutant, advantageous is yellow line. In 2D, it grows as r to the 4th, or any one of them. Let me give you the most outrageous one, this one. So we have a triple mutant with 2 mutations that are advantageous. One is neutral in 3D, so it grows as power of 10. So you have a prediction for any such law. Okay, so this is something that I enjoy, right? But it seems very useless. Let me show you some possible usefulness of this approach. So let's talk about role of mutator phenotypes in cancer. I already told you about instability. So remember, I mentioned 13% of colon cancers have mismatch repair instability. So we can apply it to that. So first, let's look at a certain mutation that occurs in a growing tumor in the absence of instability. So it grows from wild type to mutant. Okay, so these are single mutants. Or maybe the wild type can acquire a property of being a mutator, and then you have an enhanced mutation rate to create this target mutant, right? So it could be a one-step process or a two-step process through this mutator phenotype. Okay, can we compare them? So we can run simulations, stochastic simulations. And let me show you on this graph. We want to look at the number of single hit mutants and double hit mutants. So here, single hit mutants is when you create this mutant directly as a one mutational step. A double hit mutant is when you first become unstable, you have an increased mutation rate, and then you create a mutant. So here the dots correspond to simulations, stochastic simulations. And the lines are my laws that I derived, right? Remember, I can derive any power of time. But you see, the simulations stop right here. And the reason they stop is because it becomes too long. Look at the population size here. The cell number here is about 10 to the 6, 10 to the 7. Realistically, you cannot run an ABM beyond that. It just takes too long. My formula proceeds to infinity, right? So you don't need to write this, to run these very long computer simulations if you have the law. And you can see where these things cross. When the number of double hit mutants actually exceeds the number of single hit mutants. This is the size of the tumor where you can tell that this instability plays a role and it accelerates the creation of the mutants. So this is a computational tool that is extremely cheap. Instead of running a stochastic simulation, we can use the laws, and we have a prediction for the number of mutants in each of these instances. So we made it more realistic. So I just briefly mentioned. So this is microsatellite instability. As I already explained, it takes 2 hits to make a cell unstable. These are some of the genes that are involved in microsatellite instability. You have to inactivate them by a 2-step process. So you go from wild type to here, you're unstable here, and now boom, this is an accelerated rate. Can that really accelerate the process of mutation as opposed to just going directly from wild type to mutant? So that's the question. You compare from here to here, 1 step, to 1, 2, 3 steps, but the 3rd step is accelerated. What is faster? To run a stochastic simulation takes too long, but we can use these derived laws. And the answer to the question whether it accelerates it or not depends on the size of the tumor. For a very small colony, it doesn't make a difference. It doesn't even enter the picture. But as the colony expands for larger populations, you can see that triple hit mutants actually overtake the single hit mutants because of those powers of time that I mentioned before. Another application is gene amplifications that are also prevalent in cancer. Maybe I won't go into much details, but this is another example of many steps in the mutational process. And by using this new computational tool, we can calculate how many of each of those types we expect to have an expanding colony of cells. So that's it. I think I'm going to stop here. Thank my group and my collaborators. And again, looking for a postdoc, write to me. Thank you very much.
Thank you, Natalia. Other questions?
This is for my